Welcome to the Tree of Life podcast. I'm your host, Joel Ledford, and I'm here with Cassie Ettinger. Welcome,
Hello.
Cassie.
Thank you.
So today, thought I'd bring Cassie in to talk about her research and maybe ask you about how you ended up getting here at UC Davis and becoming interested in biology in general. So maybe you can start off by telling us a little bit about your background, how you ended up here, where you got your undergrad, how you became interested in research.
Yeah, so I did my undergraduate degree at UC Berkeley, and when I got there, I thought I was going to be a math major, and I was really excited, and then I decided that I didn't actually enjoy math as much as I thought, and so I started taking some of the basic intro biology classes. In my high school, I had taken some AP biology as well as AP environmental science, and so kind of in the background of my mind was something I was thinking about. And so I was there at UC Berkeley starting to pursue biology, and I thought, okay, I know I don't want to be a doctor. Blood freaks me out. Thinking about what's inside us is a little too ugh for me. And so I looked around to see if I could have a research experience. And they have a wonderful program where undergraduates can kind of apply to different projects that are available. So I applied to a couple projects, and I got into a lab at Berkeley to do some research on legume rhizobia symbiosis with Dr. Ellen Sims. And it was my first research experience, and I got to work closely with a graduate student, and then I went on to kind of have more of an independent project and worked really closely with their lab manager doing greenhouse experiments and going out to Bodega Bay, which is where I first got my field research experience, which is then I used a little bit as well for my graduate research. But that experience of kind of just getting to interact with the science and like actually, especially the symbiosis aspect, seeing how two organisms interact, a plant and a microbe, was really exciting to me. And then I got to continue a little bit of that when I studied abroad through the university study abroad program in the UK, and I worked on Drosophila, so on fruit flies, looking at fruit flies that had the genes related to human Parkinson's disease, and they were looking at how dopamine actually plays a role in vision and also in potentially in Parkinson's disease as well. And so that was very different.
Right, yeah, it's different.
It was not something I was interested in. It's still not really something I'm interested in scientifically, but it was a great experience because I got to do a full project from start to finish while I was there and like write it up and present on it. So it was very different than what I'd experienced at Berkeley. And then when I went back, I did an independent honors thesis on legume rhizobia symbiosis. And so that was kind of what got me thinking about doing research in the future as a graduate student, and specifically plant microbe research.
Wow, that's great. So, I mean, I think that's pretty common, feature in doing any kind of science. You start off on a path and you try it out, and maybe it doesn't work for you, and you have to go back and, pursue what you are really passionate about. So that's a great story. So that brings us to seagrass.
Yeah.
Right. And in reading your paper last night, I learned a whole bunch of really interesting things about seagrass, and I thought first maybe you could tell us what the big questions are in your research. Maybe explain, what was the impetus for looking at seagrass, because you're not just looking at the seagrass themselves, you're looking at their fungal symbionts. And so what are the big questions in researching seagrass? What are you interested in?
Right. So seagrass is really interesting. It is kind of similar in its ecosystem services to trees in a rainforest, right? So it's a habitat, it's a hatchery. It's also really kind of part of this blue carbon movement for, how are we going to be dealing with carbon as we move into the future? It's great for carbon sequestration because there's lots of degrading material in these seagrass beds. But it's also not a phylogenetic group. It's a morphological group. So it's all kind of three distantly or semi-distantly related lineages that have readapted to the marine ecosystem. They all have very similar morphological features. They look like grass, right? Seagrass. So it provides this really interesting opportunity to look at the coevolution of plants and their associated fungi, or even just associated microbes more generally, because now we can look at these 3 separate lineages. We can see how, what community members do they have, and then do we see convergence across the 3 groups? Do we see the same fungi with the same functions? Do we see different fungi with the same functions, or do we see different fungi and different functions? And it kind of can inform on both adaptations to the marine ecosystem on either the side of the seagrass or on the side of the microbial symbiont, so the fungi. So are they convergently evolving themselves? And then it can also inform on how we understand plant microbe symbiosis. So are there roles that are happening here that we can take away?
Right. So when I think of fungal symbionts in plants, I always think of land plants. I always think of, things like glomeromycota, the arbuscular mycorrhizae. But reading your paper, I learned that that's not common in seagrass. They don't frequently have mycorrhizae. And so, so before we maybe get into that, could you take just a moment and define for us what is a symbiosis? What's a microbiome or mycobiome in this case?
So symbiosis is hard. People define it differently. I think of it as any interaction between 2 organisms. Right? You can have mutualistic interactions where both partners are benefiting. You can have parasitic interactions where one partner is benefiting at the expense of the other, and then you can have commensal interactions where one partner benefits, but the other partner is kind of like, whatever, it's okay. So I consider all of those symbioses. Some people only consider it a beneficial interaction, but for our purposes, we'll say it's any interaction that can occur. And then kind of a microbiome would be kind of all of the microorganisms associated with a host. In our case, the host would be seagrass. And so a mycobiome, myco for fungi, is just all of the fungal community members that are associating with seagrass.
Right. So would it be fair to say that the mycorrhizae that are associated with land plants are part of their mycobiome?
Oh, for sure. Yeah, it's definitely the case. And that's a really important association as plants moved on to land, because the mycorrhizae specifically have helped a lot with acquiring nutrients that you can't acquire as easily as you could in the marine ecosystem, which may be one reason why we don't see those associations in the marine ecosystem with seagrasses moving back, is maybe they didn't need the mycorrhizal fungi to attain these nutrients. But there are other concerns when you enter the marine environment, like high salinity, and now seagrasses do have a traditional root system. They have vascular tissue, so they do have to obtain some of their nutrients through their roots. So they do have to make some of these adaptations where maybe you could see a new novel fungi coming in to form some sort of new association to provide either similar or different functions like the mycorrhizae might have been providing.
All right. So then would it be fair to say that some of the fungi associated with seagrass do some of the same functions as mycorrhizae, or no?
I think that that's a hypothesis that I would like to test further. Yeah, I do think that maybe we're seeing, especially in areas like the tropics, where there's a lot of actual nutrient limitation because they're so biodiverse that so many nutrients are being used. I could see mycorrhizae being present there. In temperate areas, I think that it's less likely. We tend to see kind of just less fungal community. The diversity is much lower in temperate areas than in tropical areas. I mean, similar to animal biodiversity. So I do think that potentially we're seeing that. We could see that.
Right, and then in reading your paper, some of those fungi are really just not known.
Yeah, well, I mean, nobody's looked at marine fungi. People think of mushrooms when they think of fungi, or they think of fungi they see at the grocery store or on their breads, molding. They're not thinking about what's in the marine ecosystem. And that's been true for traditional mycologists as well. No one, and I wouldn't say no one, I guess there were a couple scientists who looked into it, but it's not been a growing movement by far.
Right, okay. So then one other thing that was really interesting to me is that when I think of something like a human microbiome, I know that the microbiome species diversity is pretty different depending on the part of the body you're looking at. But the same's true of plants.
Yeah.
So the roots and the leaves and the stems all kind of have different fungal or microbial components.
Yeah, they do. And part of that could be having to do with the localization of certain things that they need. So you might have things involved more with nutrient uptake in the roots. And then in our case, in the leaves, we saw a lot of things that seemed related to Chytridiomycota, which are the only fungi that has swimming zoospores, right? So they may want to be more in the marine ecosystem, right, which is where the leaves are, instead of being stuck in the sediment, because most fungi are going to grow hyphally,
Right.
and not going to be moving around.
So then, I guess one of the last things I wanted to ask you about is how do you go about studying these symbionts? I mean, it's you can't really culture them in a lab effectively. So, what are the approaches that you use to study such things?
Yeah, so it's a little complicated when you start talking about eukaryotes, microbial eukaryotes specifically, and then the fungi. A lot of the work we know that's actually really kind of increased our knowledge of their evolution, especially like genomically, has actually been from culture-based studies.
Okay.
So they're still a little bit behind compared to the general like bacterial community, which is using much more of these culture-independent approaches, doing like high-throughput sequencing of metagenomes, so all of the DNA in an environment, and then kind of pulling out specific organisms. So you can kind of we're starting to move in that direction for the eukaryotes, but it's a little more complicated because of their ploidy, right? They're not haploid like bacteria. They don't have circular genomes. They can have chromosomes. So it's a little bit more complicated. So we are using lots of those genomic techniques, but we actually are relying a little bit more on the culture-based collection stuff still. But a lot of it right now is kind of, nobody really knows what the diversity of marine fungi is right now. So even before we get to what roles might they be having, we need to figure out who they are. And so a lot of that is kind of just serving with short read technology to get just some conserved marker genes so we can say, okay, who's here? And then using that information to do targeted culture-based studies where we're like, okay, so we have a bunch of Chytridiomycota, these chytrids. So people have cultured chytrids before, especially with this focus on the BD that infects the frogs and kills them, or the amphibians generally. So there are some techniques we can use to try and kind of bait those organisms out. But then again, it's just kind of increasing different methods that we could be using, adapting them from the bacterial world and kind of incorporating that more into this eukaryotic metagenomic sequencing world. Right. It's kind of the like Wild West.
Right. Yeah. So I mean, I didn't really think that, a lot of those culture independent studies are a little bit harder in eukaryotes, but that makes a lot of sense. So I guess the last question is what's next? What's on the horizon for seagrass and fungi research? What are you thinking about now?
Yeah, so we've done a lot of the kind of foundational work now, looking at who's there, and so getting more into what are they doing. So this is going to involve several different kinds of studies. Some of them are going to be experimental manipulations, right? If we grow the seagrass and then we add a specific fungal symbiont or a specific bacterial symbiont, can we see differences in the seagrass's health? And then also starting to look at differing health statuses. So most of the studies that have been done have been looking at healthy seagrass, right? We wanted to kind of set up like who's there, just generally. So now we need to challenge the seagrass. There's a pathogen, Labyrinthula. It's a heterocont. Okay. So it's not a fungi, it's not a bacteria, it's not an archaea. And, what happens when we add some of these other members of the community, when we also add this heterocont, the Labyrinthula, do we see differences in the seagrass's outcome? And so kind of getting more into this functional work. And some of that for the fungi is a little bit behind because we still have to figure out, get better genomes for these organisms, start to look at transcriptomes from the culture-based studies to kind of get that reference that isn't there for the fungi that kind of already exists for the bacteria. But just generally moving in this kind of more experimental way towards function. We could also do imaging. Imaging is always really exciting to confirm localization of some of these members and then start to look and see, do we have fungi interacting with bacteria even in the seagrass microbiome? There's also a lot of diatoms associated with seagrass, especially on the outside of their leaves. So starting to look at some of these other microbial eukaryotes and their interactions actually within the seagrass larger kind of microbiome And viruses, I'm sure, also are playing important roles.
Wow, amazing. So it's a lot more complicated.
It depends on the scale that you're looking at things. We can look at them at the gene scale, the organism scale, the community, the population. It just kind of gets crazy.
Yeah, great. Well, that sounds really exciting. Well, we appreciate you coming on today, and wish you the best of luck with your future research, and hope to see you around soon.
Yeah.
All right,
Thank you.